So kind of looking at the registration list, I think most of you kind of know who we are already, but so I'll go through this fairly quickly. We're a nonprofit and we've been doing work in the province since 1991. We primarily are seeking to improve the understanding and awareness of Alberta's lakes, reservoirs, and their watersheds. Our most widely known program is the LakeWatch program. It's a participatory lake monitoring program, and it's been in operation in the province since 1996. And in the bottom right of the screen there, you can kind of see all the lakes that we've sampled since 1996 through the LakeWatch program. We have other programs as well. Our Lake Keepers program is a kind of an up and coming citizen science lake monitoring program, and then a few other programs as well, kind of ranging from scholarships to beach monitoring, plant surveys, research, and then other education and outreach initiatives. So LakeWatch is going to be the focus of this presentation, so I'll just kind of give a quick background about the program and how we sample. Each summer we go to a number of lakes throughout the province, and we hire technicians that sample lakes with volunteers from each of the lakes. And the lake is sampled both at a profile location, kind of from top to the bottom, from the top of the lake to the bottom to get the profile information, and then samples are collected across the lake as well to sort of get a composite view of the lake. And from that sampling, we get nutrient data, other water chemistry data. We collect biological samples for algae and aquatic invertebrates, including invasive species. And yeah, we do that 4 times throughout the summer. And in the past, it's been higher, but in recent years, we've only been going to the lakes 4 times from June until September. So for the 2020 season, we were able to get to 25 lake basins. It was a very unique year in that we were able to get to all lakes 4 times. One lake we went to 5 times. Yeah, we were able to get all of that data, and there were no issues with weather or volunteers, meaning we had to skip a trip. So that's fairly unique. We had 39 volunteers that were part of the LakeWatch program last year, culminating in 307 volunteer hours. So a lot of time spent on sampling lakes by volunteers, citizen scientists, kind of across the province to support this project. And there was also a lot of financial support last summer as well, primarily from Alberta Environment and Parks, Pigeon Lake Watershed Association, Lacombe County, LICA, as well as the Moose Lake Watershed Society. Just want to recognize that we like to kind of do a shout out of our really dedicated and committed volunteers every year. And last year, Richard Normandou from Half Moon Lake was the volunteer of the year. So thanks for all your hard work, Richard. I don't know if you're on the call today, but yeah, just to kind of say we have a lot of great volunteers that we like to recognize. Last year we had 2 technicians, Kira and Ryan. They did a really great job. They had good background in field work as well as aquatic ecology. And yeah, they had a great summer, and they managed their schedules well to be able to get to all of their lakes 4 times throughout the season. Now going into a bit of the geographical spread of the LakeWatch sampling last year, we were able to get to 7 different lakes throughout 7 different watersheds in the province. So we covered a few lakes in the Athabasca, the Beaver, North Saskatchewan, just a couple in the Battle, Red Deer, Bow, and the Oldman. So a fairly good spread for lake sample last year through the central and southern parts of the province. And if you're familiar with the program, you know that we kind of have a rotation of lakes that we sample from Parks Lakes. So last year, our techs were really lucky that they got to go to the Montane Foothill Lake. So that means that we got data from Greg and Jarvis up in Switzer Provincial Park, Upper and Lower Kananaskis reservoirs in the Kananaskis country, and then Beauvais in the south. Now just going quickly into what we did for reporting, we generated 18 LakeWatch reports from all those lakes. We generally don't produce reports for the parks lakes, and some of the lakes have multiple basins, so those are accumulated into just one report. 8 of the reports have trend analysis, and we're happy to say that two new lakes had trend analysis performed. So we were able to do trend analysis at Alex and Beauvais for the first time. We also generated a summary report, and we were able to include some of the data that was gathered by Lac La Biche County last year as well. We've been in contact with the county, and they thought, and we thought there'd be some value in kind of combining some of the lake data to contextualize how the lakes are doing and comparing between the two programs. So we're excited to have that in the summary report. And we are going to be publishing a trend analysis summary report to kind of combine some of the trend results that we have. Instead of only finding it in lake specific reports, we kind of wanted to combine it a bit and kind of further knowledge in terms of what's going on in our lakes in terms of long-term trends. So now I'm gonna just get out of the presentation and show you where you can find our reports if you're not familiar with our website. So if you go to our website here, this is the main page. You go to the programs tabs, LakeWatch reports, click on that. We have a page here for a lot of different reports. So we've done bioblitzes in the past. So on top here, we have our macrophyte reports. The next table here, we have all of our summaries dating back to 2015. We have a table specific to trend analysis reports. Kind of simplified this. Before we put in every year that these lakes had trend analysis performed. And instead, we wanted to make it a bit more user-friendly and facilitate more lakes in the future by only having the most recent year report for when the trend analysis was generated. We have this is an important document to see our methods for our trend analysis, a couple of additional reports for some other programs, and then this big table has all of the reports for lakes sampled through the LakeWatch program. So lots of good information there dating back to 1996 for a few of the lakes. All right, and as Brad said, we also did a survey for some of our LakeWatch volunteers this last winter, and really the survey was to learn more about how our volunteers interact with our reporting. So lots of the questions that we asked them had to do with our figures and our tables. How do they really interact with the reports? Do they only read their own? Do they read others? Do they find value in the summary? And some of the main takeaways was that volunteers do, in fact, read our reports in entirety, and that's good for us to hear because we want to put a lot of good information in there, but still make it very accessible. Volunteers read multiple lakes reports, so they're not just interested in their own lake, but they like to explore how other lakes are doing. They've responded that the reports are generally a good mix of accessible information and advanced information. So that's important for us to know too, and that we can start to incorporate some more information that some volunteers would find really useful and interesting, while still having the reports quite accessible to people that maybe aren't as interested in some of the more detailed data. Some of the volunteers have indicated that they would prefer more figures instead of tables, and that kind of makes sense. It's easier to kind of see some data using figures. And our volunteers think that the reports are a good length. We asked them if a presentation like we're doing today would be useful, and they said that it would. Trend analysis is bringing value to their lakes, and something that we'll think about in the future, haven't done quite yet, is maybe developing a bit of a snapshot about how their lake is doing, kind of to complement the report. And really the snapshot could maybe be used by some of these groups if they want to share the results in a more palatable way to their to their network through social media. So just a few ways that we responded to the survey is that we do have new figures. So a lot of data that's usually just reported in table format that we thought would be valuable to kind of present in more of a figure format would be the ion data. So on the top right, you can see figure that we've generated for ions, and I'm going to go into that figure a bit more later. We kind of tweaked our water clarity figure. Instead of using Secchi depth, we've changed it down to EUPHOTIC depth to be able to compare how clear the lake is to how deep the lake is, or at least how deep it is in the profile location. We have developed our trend analysis reporting. Like I said, we kind of simplified the website table to kind of see just what the most recent trend is for that lake. And like I said before, we're developing a trend summary report where we combine trend results for multiple lakes into a single report and to other figures. And one of those figures at the bottom there is kind of comparing slopes of trends between lakes. And I'll go into one a bit later as well. And finally, we're doing this LakeWatch 2020 Key Findings webinar. So that's what we're doing right now. And that was one of the big takeaways from the survey. So now we'll get into some more results from the LakeWatch 2020 season. So we're pleased to say that there were no invasive mussels detected in any of our LakeWatch lakes in 2020. There were also no Eurasian water milfoil specimens detected in any of our LakeWatch lakes or other lakes that our LakeWatch program was able to sample through, just being throughout the province. So that was good as well, and we'll continue with that monitoring in this coming summer. And our LakeWatch network was also involved in supporting research done at the University of Alberta through Dr. Patrick Hennington's lab. So they collected samples from both the pelagic sampling through LakeWatch, so the composite sampling across the lake, as well as beach sites that were on their LakeWatch lakes or at beach sites that were at lakes near the LakeWatch lakes. And some of the targets that came from these samples were cyanobacteria and microcystin gene and Enterococcus, and then also a human fecal marker. So yeah, like I said, this work was mainly to support Dr. Hannington's research, but some of the samples were also able to support some of Alberta Health's recreational water monitoring. So now getting back into the LakeWatch data, this is our summary figure for average total phosphorus for our lakes. So we've got a pretty good spread, maybe a few more oligotrophic lakes than normal because we were able to get to some of those montane and foothill lakes at the bottom, and then you can kind of see the increasing phosphorus as we go up, and then a few lakes at the top there had quite a bit higher levels, average levels of total phosphorus. We decided to make a nitrogen figure as well, as that is also, like we know, an important nutrient for algal growth and productivity. Looks a little different from the phosphorus. More lakes are kind of sitting in that hypereutrophic class, which kind of makes sense considering the abundance of nitrogen in our lakes here in Alberta. And then another interesting one that we always like to present on is chlorophyll. Kind of follows more of that total phosphorus figure, but kind of a pretty large range as well, with one lake maybe kind of sitting quite a bit higher than others. But yeah, you can kind of see we had a pretty good spread as well in terms of algal productivity. And then we also generate a summary figure for water clarity, and kind of going with that theme of going with EUPHOTIC depth and being able to compare that to the lake profile depth. We can see the variation through our LakeWatch lakes in terms of their water clarity compared to how deep the profile location is. And just a few interesting things that we can kind of take away from this figure when we combine water clarity with the lake profile depth is lakes like Jesse and Alex and Lacombe, they're unlikely to be able to get into the you know, the mesotrophic or oligotrophic range just because of how deep they are. So the figure kind of shows that most of the lake throughout the season was getting light to just about the bottom of the lake. So it's kind of an interesting takeaway from being able to visualize water clarity with the lake profile depth. When we look at how the lakes were for average microsystem throughout the year, a lot of lakes were kind of below detection in terms of microcystin, which is a toxin produced by cyanobacteria. And then levels gradually kind of increase for lakes that are a bit more productive. But none of the lakes had average levels that were greater than that 20 microgram per liter threshold. That's kind of a threshold for health risks for aquatic life and for humans and terrestrial animals as well. And yeah, we've kind of heard that guideline might be changing. So we're interested to see how that will play out. And one thing that we always try to take away from this figure as well is when levels kind of get, you know, above 2 or you know, like they're quite a bit above the detection limit, then that, because this is a representation of the average levels across the lake, that might mean that certain areas on the lake where there are accumulations of cyanobacteria might actually be above that 20 microgram per liter. So we always kind of present this figure with an asterisk that says, you know, like if you're even though the levels are lower than 20, that doesn't mean that at specific locations those levels are still going to be below 20. So something to keep in mind when it comes to microsystem reports. The figure just tries to summarize the lake ion chemistries by watershed. A few things that we've taken away from this figure is that calcium at our lakes is fairly conservative. So this figure, I'll just say the y-axis is on a log scale to be able to kind of capture that the range of ion values between our lakes to be able to see the spread a bit better. But yeah, one thing that we've noticed is calcium seems to be fairly conservative across our lakes, at least the ones that were sampled last year. And bicarbonate seems to be the dominant ion, which I think we already knew, but that it also appears to be fairly conservative between our lakes. And kind of as expected, the pink and the blue, green, and yellow, those are lakes that are from our Bow, Old Man, and Athabasca watersheds, and those are lakes that were you know, in our montane foothills. So they had proportionally lower levels of ions, where the other watersheds had quite a diversity in ion levels and distributions. So now I'm just going to go into that ion figure that I talked about. Is a new addition to the LakeWatch report. So it's we've kind of made it as a split figure where the top has just a standard y-axis, and we have each of the major ions on the x-axis, and the bar shows the average level of that ion throughout the season, and then the bar range here is actually just the range of values throughout the season as well. So it kind of gives us an idea of the relative proportion of these ions throughout the season, as well as like how they vary. So for Beauvais, there's very little variability in most of the ions, except for maybe bicarbonate. There's been some variability throughout the season. And then the bottom figure is very similar to the one that I showed in the last slide, where we have all of the lakes kind of in a single figure. And just to highlight the lake that we're reporting on, we've made it the blue. So we can kind of see the Beauvais in relation to the average or the other lakes that were sampled throughout the season have proportionally low levels of kind of all of the ions. And then kind of compared to, say, Jesse Lake, where we know has higher ion levels, we're able to see that some ions have lots of variability throughout the season, and that it compares kind of higher to other lakes that were sampled through the LakeWatch season by having a blue line that is quite a bit above the other gray LakeWatch lakes. It's also, the figure gives us the ability to kind of identify maybe some unique characteristics in the ions for lakes. So for Lac Santé, it has proportionally high ion levels, except for calcium is proportionally low compared to the other LakeWatch lakes. I believe it still has high alkalinity, but yeah, it has lower calcium, free calcium compared to other LakeWatch lakes. And then the East Basin of Mayatin is unique as well in that it was the only lake sampled last year that had sulfate levels that were actually greater than bicarbonate. Otherwise, all lakes, the predominant ion actually was bicarbonate. All right, now to some more anecdotal sort of observations that we had through the LakeWatch 2020 season. So one thing that we noticed was at the end of June, we had volunteers at Lac Santé and WABAMUN reach out to us and say, hey, we have these pretty significant algal growths happening at our lakes, and they're impeding our ability to access the lake. And obviously the first thing that they're worried about is cyanobacteria. But kind of looking at the pictures and receiving some samples from these lakes, we were able to confirm that they were in fact green algae blooms. And one thing that we kind of say to our volunteers to be able to differentiate the two is that the green algae has a bit more structure to it, like you can see in that picture from WABAMUN. The volunteer is able to hold the colony, and it has much more rooted-like structure. And you can't do that with cyanobacterial blooms. So that's one way that we're trying to teach our volunteers to be able to differentiate the two. And that's important because green algae isn't toxic, but it still can bloom and impede recreation. But then another thing that we notice is that at those two lakes in particular, while the green algae blooms kind of happened towards the end of June, as the summer progressed, both of those lakes started to develop fairly significant cyanobacterial blooms, kind of different types. You can see the one at Lac Santé, it was more that Aphanisomenon, or more green cyanobacteria, but at WABAMUN, it was a brown looking Gliochichria dominated bloom. So it's one interesting observation last year to maybe see a transition from a green algae bloom to a cyanobacteria bloom, and it's something that we're going to be maybe looking out for more in the future to see if that's a trend for these lakes or for other lakes in the province as well. Just going to spend a little time on Mayatin as well. It's a unique system in that we sampled the basins separately. The lake was first sampled by LakeWatch in 2011 as a single lake, but from that sampling season, we learned that the West Basin is quite a bit deeper and had fairly different water chemistry than the East Basin. So then in 2012 and 2013, the basins were sampled separately. And in those years, the West Basin had proportionally lower levels of total phosphorus and chlorophyll A, while the East had higher. And then there was a bit of a break in sampling, and then we sampled Mayatin Lake again in 2020. And interestingly, the basins now are more similar. We think that it's because water levels have come up in the last couple of years with having more precipitation. The picture there kind of shows the connection between the two basins, and before the lakes weren't, you weren't able to put a boat through that through the narrow, but 2020 we're able to. So it's kind of interesting to see with water levels returning to Mayatin to see the basins having more comparable water quality than before. Skeleton Lake is a bit of a similar case where there's two different basins that we sample. So we have the North Basin that we sample, and then the South Basin that we sample. We've done a lot of sampling at Skeleton Lake over the years, and kind of in the past 2 years, we've noticed, and this has also been through a volunteer that lives out there, we've noticed that the North Basin has become proportionately a bit more turbid, or in the summer there's been more productivity, although the productivity is more of like a brown cyanobacteria. We think that it's a species in the Planktothrix genus, either Planktothrix agardii or rubescens, that kind of unique cyanobacteria that may live further down in the water column. And we've actually learned that at Skeleton North, that the Planktothrix is just as abundant in the winter as well. So kind of an interesting situation at Skeleton, and that yeah, in 2020 we saw quite a big difference in water quality between the two basins. And in that satellite image, you can see that the south is proportionally clearer and the north is proportionally a bit more turbid, and the phosphorus levels are quite different between the two basins as well. And then we also know that the microcystin is quite different between the two, and that Skeleton is one of the higher lakes we have for microcystin, and that's kind of to be expected with Planktothrix as being a microcystin producer. But then Skeleton South proportionately lower levels of microcystin. So it's interesting to have a lake system like this with very, very different levels in terms of cyanobacteria toxins. And then maybe just to kind of contextualize the differences that we're seeing at Skeleton, you can see the satellite image at the top there of Skeleton from August in 2017. It was actually the South Basin that had a bit more productivity, and the North is a bit clearer. And the Secchi readings confirm that a bit as well, that the North Basin had high variability in water clarity, but that it was on average more clear, and that in more recent years in 2019 and 2020, we've seen kind of a reversal where the South Basin has become proportionately clearer and the North has become a bit more turbid, and that trend kind of holds for phosphorus as well. So we're talking a lot with the volunteers out there and learning more about the lake, and we anticipate that some of the differences have to do with water levels, as well as the morphometry of the lakes in terms of the North Basin is very deep, it stratifies, it doesn't mix in the summer, where the South is much larger, it's proportionately more shallow, so it does mix. So we're curious to see if some of those differences that kind of explain why we see differences in water quality, and how those might change as water levels return to the lake, or if we end up having a drier summer again, if the narrows eventually become more separated, as in 2020 you're able to navigate between the two. We're curious to see if the basins will continue to kind of diverge, or if they will kind of follow their similar trend in the past, where they were a bit more similar in terms of water quality. With the WABAMUN Lake, one key observation that we had is that it had the highest total phosphorus values on record. So this is the this figure kind of shows both chlorophyll and total phosphorus values going back to 1980, 1981 for both of the parameters, and that it was a unique year in that phosphorus levels, for whatever reason, seemed to be very, very high. And one of these sampling events, we even had a QC that confirmed that the levels were that high. So kind of an interesting thing for WABAMUN. We're not quite sure as to why there was such a large spike, but we're curious to see how the total phosphorus will play out for WABAMUN in the future. One other thing that I'll point out, and I mentioned earlier, is that in our summary, we integrated some data from Lac La Biche County. We did it kind of in a table format, just because the sampling program differed a little bit, and that a lot of sampling occurred in the Lac La Biche County lakes in June, proportionally lower in July, and then more in August. So we ideally we wanted to include them in our summary figures, but just because of a difference between the sampling programs in terms of the frequency, we thought it would be better to represent the lakes in table format. So you can find this table in our summary report and see how some of the Lac La Biche County lakes, including Lac La Biche, compared to our lakes that were sampled through LakeWatch last summer. All right, so now I'm going to go into some of our trend analysis work. So just as a bit of a primer, so how we do trend analysis is that we need at least 10 years, more than 10 or more years. I can see my symbol there is wrong. We need 10 or more years of data for a lake to do trend analysis. There can't be a data gap that is greater than 1/3 of the total length of the sampling history. And then we need a consistent seasonal sampling effort. So ideally between June and September. We also remove May and October data because generally LakeWatch data sampling only occurs between June and September, although sampling at some of these lakes has occurred as well in May and October. There are some exceptions to this for WABAMUN. There's more data that has May and October data included than not. So WABAMUN is the only lake where we actually have included May and October data, just because there is so much of it in the historical sampling. And then we only select 1 day per month if more than 1 day has been more than one sampling event has occurred in each month, and we always select the day closest to the 15th of the month. We evaluate the seasonality of the data in terms of if there's differences in seasons. We indicate the month as a season. So if there is a seasonality or differences in trends between June's, July, August, or September data, then we go with the seasonal Kendall test. If there isn't seasonality in the data, then we go with the man Kendall. And then our results indicate a monotonic or unidirectional trend for total phosphorus, chlorophyll A, total dissolved salts, and Secchi depth. And then we also can kind of get a rate of change or a slope for some of these trends as well. And another part of our trend analysis and reporting is to generate box plots and line figures to sort of visualize better what some of these trends look like over time. And as of this season, we have done trend analysis at 16 lakes sampled through the LakeWatch program. And maybe I'll just clarify that data that goes into the trend analysis isn't just through the LakeWatch program, but if lakes have historical data that was generated through Alberta Environment and Park sampling, that data is also incorporated into our trend analysis. So these are a few of the figures that we're going to be presenting or publishing in our trend report. So the top there just shows the range of trends, or the range of years that we've used for these lakes to have trend analysis. So there's a bit of a variability. Some lakes that have been sampled by the province for a long time have a larger range, such as WABAMUN, Silva, and Moose Pigeon. And then a few other lakes that have just had a really consistent sampling effort through our LakeWatch program, such as MINNIE Lake, Laurier, even Crane Lake, have shorter durations, but just because they're sampled just about every year, we are able to do trend analysis on those lakes as well. The bottom figure here just shows how many lakes are either significantly increasing or decreasing or have no trend for each of the parameters. So interestingly, for chlorophyll A and Secchi depth, the results kind of jive there, where we would expect if only 2 lakes, say, are significantly increasing in Secchi depth, it makes sense that chlorophyll is decreasing, and that an increase in Secchi depth means a decrease in water clarity, primarily due to productivity. Kind of a large finding from a lot of our trend analysis work is that a lot of lakes are significantly increasing in their total dissolved solids. That's not to say that some lakes are also decreasing. And then total phosphorus, kind of counter to what you might expect, more lakes are actually significantly decreasing in total phosphorus as opposed to increasing, although there are many lakes that have no significant differences in total phosphorus over time. So this is that slope figure that I talked about earlier. So This figure just incorporates the slopes for each of the parameters into box plots for us to kind of get an idea of the range in change in some of these parameters at lakes that have significant trends. So that's just to say that the data that's incorporated into this figure is only for lakes that have statistically significant increases or decreases. So like I was saying, for chlorophyll and seke, it kind of makes sense the way that they've sorted out. Although chlorophyll, there's quite a range in terms of how the magnitude of change that we kind of see for lakes that are increasing. And without going into this too much, because I know we only have so much time today, I'm just going to focus on a couple of the outliers because there are a few interesting cases. So these are lakes that are kind of bucking the trend of how other lakes have, how their magnitudes of change are. So MINNIE Lake and Alex Lake. So MINNIE is a bit of an interesting case in that we're seeing significant increases in chlorophyll and decrease in seke depth, although total phosphorus seems to have been decreasing over time at MINNIE. That probably is more driven by a change from the past, although in recent years we kind of see potentially phosphorus increasing at MINNIE, but that's an interesting case that we're gonna be keeping an eye out for. Is the trends at MINNIE Lake seem to be fairly unique compared to some of the other lakes. Alex Lake has the greatest slope or rate of change of total dissolved solids, and we're curious if that has to do with the diversion of the Red Deer River, in that the diversion ceased in 2010, and kind of since then we've seen an increase in total dissolved solids at Alex Lake, so that may make sense. Although I'll just kind of say as a disclaimer, there's a bit of a data gap in that period of time, so want to kind of have a just kind of see that trend with caution in terms of not having the data right around where the diversion actually was last operational. Other interesting trends that we're seeing in terms of total dissolved solids is that WABAMUN and moose have very steep rates of change for total dissolved solids. And one of our first things we want to look to sort of understand maybe where this is coming from is, oh, why is my slide not going? Oh. Okay, I think we're good. Yeah, so one of the things that we first kind of look to is to see how the total dissolved solids, their trend compares to lake levels, and if that shows up again. For WABAMUN, when we look at the lake level trend over time, and I've just indicated the period of time that compares to our trend analysis, at WABAMUN, it doesn't really seem to make sense that the total dissolved solids is directly related to lake levels, at least upon first glance, and that through the trend analysis period, we've seen a relative decrease and then an increase again in water levels. But through that time, the total dissolved solids have been steadily increasing at about 4 milligram per liter per year. Moose, it's possible that the total dissolved solids are more related to lake levels, and that a recent decrease actually at moose may be related to a recent increase in lake water levels. And in the next figure, there is a bit of a data gap around a time where we can see the lake water levels having a spike. So yeah, it's kind of interesting to see that there was quite a large increase in total dissolved solid levels at moose, but that it's possible that the total dissolved solids at moose is more related to lake water levels. Just want to point out our trends at Beauvais, and that it's a bit of a unique system in that there have been significant decreasing trends in both total phosphorus and chlorophyll over time, and that it's one of our best cases of where those two have tracked fairly well together, and it goes back to the 80s. So it's interesting to have done that this year for the first time, and to see that yeah, that those are tracking very similar together in a decreasing fashion. And lastly, I just want to point out for the trend analysis as well that even if there are no significant trends, that's still useful information to say for a long period of time, like at PIGEON, that there are no significant increasing or decreasing trends for total phosphorus or chlorophyll A. Although looking at the figure for chlorophyll and total phosphorus, we've observed that there's a large variability, maybe a larger variability in recent years compared to previous years, and that's potentially why there's no unidirectional trend. Is that just recent years at PIGEON there might be a bit more variability in these levels, and that some years there are very high levels of chlorophyll, and that's not to say that maybe earlier in the year the levels are still low. So, and then also interestingly, we can kind of see at PIGEON that there is a significant increase in Psyche depth or water clarity, and that's also probably related to sort of the increased variability in recent years, sort of driving that relationship. All right, so sort of wrapping up what data we thought is interesting to look at from the last season. Looking ahead for LakeWatch 2021, we'll be getting to 26 lake basins, so a very similar effort as last year. We have hired 2 techs again, LakeWatch techs, and they'll be starting on Monday, and we'll be doing 2 weeks of training, and then they'll start to sample lakes in June. Just like last year, we'll continue with our invasive species monitoring through the LakeWatch program. So we'll be monitoring for Eurasian water milfoil as well as dry sanded mussels. The LakeWatch techs and the LakeWatch program will be supporting the beach sampling rec water monitoring program, actually that Alberta Health is doing this year. So they've kind of brought us into the network and to be able to support some of their rec water monitoring, and that we'll be getting beach samples from our LakeWatch lakes for cyanobacteria and enterococcus to help them in their effort. And then finally, I'll just point out that there's going to be a monitoring buoy deployed in Pigeon Lake this year, and that through the LakeWatch sampling, we'll be able to support the validation of the data that's being collected at that monitoring buoy Pigeon. All right, and with that, I will wrap up, and yeah, just please let me know if you have any questions about our LakeWatch season in 2020.
Okay, thanks, Caleb. It's obvious it's hard to draw any like broad conclusions because all the lakes tend to be doing different things, and it's very specific to each lake, kind of how it's behaving. But you did a good job trying to capture some of the interesting things happening out there. Are there any questions? You can type them in the chat, or you can turn on your microphone and your camera if you're comfortable and ask them directly.
Hi, it's Pauline calling in from Buffalo Lake. Last year we had some testing done there. We believe it was by the University of Alberta and a pilot project. So I'm not sure if that was, you're referring to one of your presentations, Dr. I don't know if I catched his name. Yeah, Hannington, I think you said. We've been trying to find some information regarding the testing that was done there. They did find human fecal matter. The beach was closed with some very poor signage for a period of time. And it's all very vague. We don't have a lot of information. People are quite, interested in pursuing this, finding out more information. So I'm not sure if I, got in touch with you and you would be able to help us out to find more. You did mention that this year they will be doing some Alberta Health Services, some beach sampling. So how would we find out if Buffalo Lake is on that list?
Yeah, thanks for your question. There was, I believe, yeah, Buffalo had a few sites that were sampled last year through the EDNA program, and yeah, you definitely follow up with us. We can kind of maybe get you in touch with Patrick and to sort of understand a bit more of the data that was generated from those sites last year. As far as moving forward, there are priority sites on Buffalo, to my knowledge, and that if the operators are being solicited at those sites to see if they're going to be able to sample through the network. And then if they can't, then it's actually our network that's going to be able to sort of fill in the gaps. So yeah, we can also kind of talk to you about that on the side to see just maybe for some of the areas that you're concerned about, how they're actually going to be covered at Buffalo Lake this coming summer.
Great, that would be perfect. And I could just call in on the number that's on the screen right now.
Yeah, or email. Yeah, either works.
Okay.
Yeah.
Perfect. Thank you.
Yep.
Just a question. That's Ray Makowecki, Caleb, and Brad. Just wondering if you've noticed any correlations between flooded vegetation like willows and increased higher water levels recently, and the differences that might be occurring in the water quality with regard to some of these parameters. So looking at flooded willows and aspen that are decaying because of the flooded waters, are you seeing any kinds of trends, or will your data maybe show some of those trends of flooded vegetation? I particularly reference a lake like Vincent Lake north of St. Paul that has winter killed 2 years in a row, fish-wise, and it's flooded big time with willows that are now decaying, along with aspen that have been flooded in the last 3 or 4 years. So I'm just wondering if the trend data can show anything with regard to those types of lakes that are actually higher water level, but because of the decay that's occurring of those woody types of vegetation, whether we're getting a difference in water quality.
I can take a stab at that, Caleb.
Yeah, okay, go ahead.
Oh, Caleb, I think your headset's not working again.
Can you hear me now?
A little bit better. This happened. It's okay. Don't panic, anyone. This happens every time Caleb and I have a virtual call.
Can you hear me better now?
You sound quite quiet.
I'm gonna log out and log back in, but yeah, take the question and I'll be back.
Okay.
I mean, you can answer it later, Caleb, but I just was throwing it out since you're on the screen.
Yeah, I mean, I can try to answer that. The data that we have probably wouldn't be specific enough to be that diagnostic in terms of, is it the decaying vegetation or not? I know, for example, though, at 2 lakes where we had increasing water levels, Moose Lake and Laurier Lake, in those cases, what seems to be happening is that bays that were once kind of disconnected from the lake and bays that are quite shallow have a lot of macrophytes and tree growth. Once the water levels went up, those bays became much more strongly connected to the main basins of the lake. And I'm really curious to know if there is actually any impact from that water entering the lake. And it's really hard to say. I know at Laurier Lake, there seemed to be a strong decline in water quality with an increase in water levels. And we do know that there are trees around the shoreline that have been dying because of that. So it's an interesting idea. I just don't think our data is that diagnostic.
Okay, thank you.
Caleb, do you want to try talking again?
Yeah, I think I heard you talk about Laurier Lake, and I think that's what I was going to talk about as well, is that we don't have too many lakes that have enough data say that we're able to see that at a lot of lakes, but that Laurier is an example where with increasing water levels recently at the lake, we are seeing increase in total phosphorus and chlorophyll, and it could be related to some landscape characteristics, but that yeah, like Brad said, I don't think we can sort of
identify reed beds or willows or anything. But that I think like relating water level changes to our trends would be interesting to kind of look more into in the future.
Okay, could I have one other question? Okay, the question is with regard to Moose Lake. There's a plan to remove the control structure that has existed on the outlet of Moose Lake. And looking at some of the evidence and information from some of the water quality work, there is concern that during particularly the low water years, there will be more algal growth, and because the storage supply during those high wet years will be reduced, therefore those low water years will be more severe, and we'll probably end up getting increased algal growth. Would you like to comment on that? So if you take out a control structure that gives you more storage during wet years, if it was removed, then we expect that the low water years are going to be actually lower because it will not have stored as much water. Do you expect that the algal growth and the water quality will be reduced, and maybe the beach areas again will be of poorer quality because of that?
It's a good question. One of the Moose Lake Watershed Society directors sits on our board, and last night at a board meeting was kind of the first time I heard about some of the discussion up there around the removal of the control structure. So I won't comment just yet because this is the first I've kind of heard about it. Maybe the more recent or the really recent phosphorus budget, nutrient budget could give us some indication of the types of impacts we might expect. But yeah, I can't comment just yet, but just know that we are in conversation with the Moose Lake Watershed Society, and they do sit on our board of directors as well. So it will be a conversation we have going forward.
Please share that information, Brad or Caleb. It'd be useful for us to hear from some of our organizations out in the Northeast. Thank you.
Great, thanks for the question. Any questions from anyone else? I don't see anything in the chat. And you're welcome to turn your mics on if you want to ask yourselves.
Yeah, I have a question just regards to the trend analysis you guys have done is really interesting. And it's really kind of cool to see how different lakes have responded to the different environmental variables. Is there any plans going forward to try to see if we can kind of do more explaining of why we see particular trends in particular lakes? Or is that kind of beyond the scope of what you guys are planning on working on? Like, is there certain types of lakes that have been tending to go this direction with, you know, say when we look at calcium going up versus, you know, chlorophyll A, you know, all these different variables you were talking about to put it into a more complete picture?
Caleb, do you want to take a stab at that?
Yeah. I think it would be really valuable to sort of maybe explore some of the more striking trends. Say one of the ones I presented was for WABAMUN, and that we've had conversations with the group out there to sort of maybe sort of dig into that a bit more to see, you know, which ion that's part of TDS, or like which ions in particular are increasing more relative to others, and then can we relate that to some of how the lake is being managed, and that's something that's not really within our wheelhouse right now, but that we would like to support moving forward. But yeah, you could explore a lot of different relationships, and for our trend analysis, we sort of see value in being able to identify the trends first, and then to work with groups and even other scientists to sort of explore maybe why some of those are happening, but that we don't have the capacity to do a lot of the diagnostic and additional analysis that would be required to sort of understand more specifically why these lakes are behaving the way they are.
I would say too, the trend analysis summary report is a good step in that direction. We've thought about, looking for sort of regional trends, but neighboring lakes often can behave very differently from each other. And so I think it is often a case of each lake has kind of very unique factors that are contributing to the trends. But, at some lakes, they're asking us about investigating road salts in terms of increasing TDS. So, we're looking at that. Water level changes impacting TDS, of course, or like I said, with lakes like Laurier and Moose Lake, changes in water levels impacting nutrients, another interesting question. So at this point, it's kind of a case-by-case basis, though. But it's a great question, Michael, and definitely something that we'll keep in mind, because I know you're up in the Lac La Biche region, and there's probably questions about trends in Lac La Biche as well.
Yeah, thank you very much, guys. Yeah, it's interesting to see, the high water levels seem to have increased nutrients in some lakes and decreased in others. So could we make some generalizations about which lakes we would expect that to happen in versus not? And, and yeah, anyways, I guess each of these reports could end up becoming a plethora of studies in and of themselves, trying to find some explanations. Thank you, guys.
Okay, so we are at 1 o'clock now, and we had just set aside an hour for the conversation. So I think we'll wrap up. Just a reminder that the session is recorded. So once it's posted, feel free to share it if someone's missed it. As well, feel free to get in touch with us. Caleb had our contact info up on the screen there. Caleb, do you want to throw that slide up if it's not too much effort?
Yeah, I just have to open my presentation again.
Oh, sorry. I'll put it in the chat. I'll put our contact info in the chat for now. And everyone is welcome to reach out if you have further questions about your lakes, or after you read the reports. If you have more questions, then don't hesitate to reach out. So anyway, thanks for everyone who joined us today. And I'll just leave the session open for a couple more minutes here, but I'll let everyone get back to their day. So thanks everyone, and thanks to Caleb for talking for so long. I really appreciate it. You did a good job. Okay, I'm going to stop the recording.